Welcome back, friends. Welcome in another video of complement pathway. This video will going to be the fourth video lecture of lectin pathway. Fourth video lecture of our complement pathway, and the topic of this video is the lectin pathway. So first thing, please go and watch the third video, that is about the classical pathway. If you are new in this channel, then it will be helpful for you to understand about the concept of lectin pathway. And otherwise, if you know about classical pathway and here you have come to learn the lectin pathway, then let's start. So in the second video lecture, we have talked that complement systems have three different pathways, via which they can perform various type of functions, right? That will ultimately complement antibody, right? And that is why they are named as complement proteins. So in the classical pathway, we have seen that the entire pathway will be activated by antigen antibody complex, mainly the Fc portion of the antibody. When Fc portion of the antibody bind with complement, it can lyse the antigen that is present at the FAB end of the antibody, right? So here the things are different. Here antibody will not going to play the role of activator. So as here antibody is not going to play the role of a activator, that is why we are considering this pathway as a part of innate immune defenses. Very important point for your MCQ exams. That means if they ask that which pathway of complement is a part of innate immune response, so you have to tick on lectin pathway. So now one thing is clear, that here antibody will not going to play the role of activator. Then which one will going to play? So here the lectin protein will going to play the role of activator. So now it is important to understand about what lectin is. So lectin here is a protein, more specifically saying a acute phase protein. So what this acute phase protein do? This acute phase protein will going to recognize or will going to target a specific carbohydrate. So as I have mentioned here, a specific carbohydrate, that means it will going to target only one type of carbohydrate. It will not going to react with any other type of carbohydrate. So here the carbohydrate is mannose. So we have learned that lectin will going to play the role of activator. So now the question is, what kind of bacteria are at risk from the lectin pathway? So the bacteria who are having, although bacteria are either spherical or rod shaped, but here I am taking a circle to designate bacteria. So bacteria who are having mannose residue in its surface, or more specifically saying who have terminal mannose residue on glycoprotein or polysaccharide present in the surface of the bacteria, can be destroyed by lectin pathway. That means any bacteria that contains glycoprotein or polysaccharide that have terminal mannose residue can be destroyed by lectin pathway. So that is the thing. So any bacteria that contain terminal mannose residue containing glycoprotein or polysaccharide on their surface can be destroyed by lectin pathway. And we have also learned here the lectin will going to play the role of activator, and that is why we will not going to consider lectin pathway as a part of innate immune response, as a part of your humoral adaptive immune response. Instead we will going to consider lectin pathway as a part of innate immune response. Now let's talk about the how it will be accomplished. So first of all, bacterial protein and polysaccharide with terminal mannose residue. Okay, first of all, MBL will going to bind with that bacterial protein and polysaccharide terminal mannose residue. So MBL here is mannose binding lectin. Okay, so this mannose binding lectin will bind with the terminal mannose residue. Okay, and then upon this binding there will be two protein that will be activated, MASP1 and MASP2. Now the question is, what stands for MASP? So MASP is mannose activated serine protease. Okay, mannose activated serine protease 1, mannose activated serine protease, mannose associated serine protease 1 and mannose associated serine protease 2. Okay, so this we have also talked about serine protease in case of our classical pathway, if you can remember, right, that C1R and C1S was our serine protease, right? So here those serine proteases are MASP1 and MASP2. Okay, clear? So here MASP2 structurally resembles C1S. Okay, so this MASP1 and MASP2, mainly MASP2 will going to cleave C4 and C2 and activate them, because we have talked about that thing that complement proteins are usually in a proenzyme or zymosin, and they can be activated by proteolytic enzymes. So here the proteolytic enzymes are MASP1 and MASP2. First, C4 will be cleaved into C4b and C4a, and I have told you that C4b will be the larger fragment that will going to take part in further reaction, and C4a will be dissociated. So here C4a are dissociated. Okay, then C2 will bind with that, and it will be again cleaved into two parts, C2b and C2a. So here C2a is our larger fragment that will going to take part in further reaction, and C2b will be dissociated. Okay, and as a result we will get our C3 convertives. Okay, so although this is not the end of that pathway, but I have told you that we have also wrapped our classical pathway video after formation of C3 convertase. Here we will also going to stop the video after the formation of C3 convertase, and again in alternative pathway we will going to talk only up to formation of the C3 convertase, because the rest of the pathway are same for both all the three pathways. Okay, so we will going to talk about this in a separate video. Okay, so this is the thing. So for a quick recap, here the activator is the lectin, and lectin will recognize terminal mannose residue present on the glycoprotein and polysaccharide that are present on the surface of the bacteria. Okay, first MBL, that is mannose binding lectin, will bind with the terminal mannose residue, and as a result MASP1 and MASP2, those are serine protease, will be activated. Okay, and this serine protease will going to cleave C4 and C2 into C4b and C4a and C2b and C2a. Okay, and C4b and C2a will going to take part in further reaction, and C4a and C2b will going to be dissociated. Okay, and as a result the formation of C3 convertase occurs. Okay, so that is the thing. Now another important thing, those type of terminal mannose residue whose presence is important for the binding of lectin and for the activation of the lectin pathway are not present on the surface of the mammalian cell. Okay, and that is why lectin pathway of complement activation may be responsible for discrimination between self and non-self. Self and non-self means which protein is the body's own protein and which one is not. Okay, we will going to talk about self and non-self in details when we will going to talk about the autoimmune disease. It will going to be a very important concept. Okay, so In this video, if I try to summarize the entire points, so lectin pathway is not activated by antibody. That is why here we are taking lectin pathway as an example of innate immune response. So here lectin will going to play the role of the activator. Okay, lectin will recognize terminal mannose residue present on the surface of the bacteria. So any bacteria having terminal mannose residue in its surface are at risk with the lectin pathway. Okay, so upon binding of the mannose binding lectin, two serine proteases named MASP1 and MASP2 will be activated. Okay, and upon their activation, they will cleave C4 and C2 into C4b and C4a and C2b and C2a. So C2b and C4a will be dissociated, and C4b and C2a will take part in further reaction that will ultimately forms the CT convertase. Okay, and the last point was, as mannose residues are not present on mammalian cells, so lectin pathway have the ability to distinguish between self and non-self. Okay, so I hope this video will be helpful for you. Thank you.